post-mortem biochemical analysis is generally the medical examination of a dead body to discover the cause of death. So post-mortem chemistry, it plays a very significant role in forensic pathology and in biochemical analysis of the vitreous humor, cerebrospinal fluid, the CSF, blood and urine are very much important when we want to determine the cause of death, or in a situation whereby there are forensic cases. Forensic cases must not only be where there is death, because sometimes there might be perpetration and we want to determine whether the perpetrator left some trace evidence behind. So the post-mortem interval is the time that has elapsed since death and used to estimate this time interval. For example, we can use the vitreous humor analysis. The vitreous humor is a 4 to 5 millimeters of colorless gel, and it is found in the eye, and it is resistant to some post-mortem changes due to its location and inert nature. You know, the eye is usually one of the biologically privileged areas where most pathogens cannot access. So we can use the vitreous humor in case we want to determine the time of death. So it is useful as a source of DNA or in diagnosis of a disease. So we can use it to obtain our DNA from the vitreous humor, or in case we want to diagnose the type of disease that an individual died of. So it has some contents like electrolytes including sodium, potassium, chlorine, calcium and magnesium, and their concentration are measured using various analyzers, and their concentration is related to the time since the death took place, using of course various equations. These different results result due to maybe things like the temperature of the area, humidity. Environmental factors generally affect the vitreous humor analysis and type of death that is associated with that. So at a higher temperature, the concentration are less stable and the degeneration of the sample speeds up. When the temperature is high, of course, the degeneration of the vitreous humor will be higher, and the time in which of death will speed up. As the time of death increases, the potassium concentration in the vitreous humor rises and the sodium and calcium concentration falls. So it means we can use actually the amount of potassium in the vitreous humor. If it is so high, we can measure it using a specific equation to determine the time in which the death took place. And also we can use sodium and calcium concentration, since they fall as the time of death increases. If they are too low, we can maybe gauge using an equation the time of death that the individual died. The ratio of potassium to sodium decreases linearly with time. The reason that potassium level rises after death is because of a leak in the cell membrane that allows the concentration to reach equilibrium with the potassium levels in the bloodstream and plasma. So since there is a leak in the when there is death, there will be a leak in the membrane of the vitreous humor. So the potassium level, since it is always low in the vitreous humor, it will try to reach an equilibrium with the potassium level in the blood plasma. So as a result, the potassium levels will be rising higher. So it is a good estimate used to determine the time of death. So there is also CSF analysis. CSF in full is called the cerebrospinal fluid analysis. So it is a cellular liquid that provides a barrier to absorb shock and to prevent injury to the brain and the spinal cord. There are several substances in CSF that can be measured, including urea, glucose, potassium, chlorine, sodium, protein, creatinine, calcium, alkaline phosphates, and cortisol. Concentration of these substances can be estimated, and this information can be used to determine the time in which the individual died. For example, A higher concentration of urea indicate kidney disease, or from what type of disease this person died. For example, a high concentration of urea may indicate kidney disease. High levels of cortisol, the hormone released under stress, could indicate violent death. Creatinine is stable post-mortem, so the concentration at death is preserved. So the creatinine is always stable, so the concentration at death. Remember, creatinine is produced as a result of your action of the muscles. So when you die, it means there will be no more production since your muscles won't be moving. So creatinine, it is stable in postmortem, so the concentration of at death is preserved. Sodium and potassium can be measured in CSF to predict the time since death, but it is not accurate as it will be when we are using the vitreous humor analysis. So we can also use sodium and potassium to measure the time since death, but it is not so much reliable as we could be reliable using the vitreous humor analysis. So we can also do toxicological analysis, which involve the measurement of the concentration of drugs and toxins and substances contributing to death. For example, somebody took some poison, or somebody maybe was injected with some poison. So we can do toxicological analysis to determine the cause of death. It is done by comparing the concentration to the lethal limits. Remember, there is a lethal dose in which when somebody takes a certain amount of the dose is what is most important. Where somebody takes a dose of a certain amount of something, it leads to death. So the most common samples used include blood and urine. Samples are put through various tests and instruments used commonly. The most commonly used instrument is the gas chromatography mass spectrophotometry. So there is also, we can also analyze the blood and urine analysis. When blood is used for toxicological testing, drugs are usually target of analysis. The toxic substance in blood can be found, but urine, kidney, and liver only metabolite may be found. This is because of different retention time. So we can find the toxic substances in the blood, but when we are using urine, the kidney and the liver, we only go for the metabolite that they produce. This is where we can find the toxic substances. For example, cocaine can be detected in the blood for 2 to 10 days, while in urine it is detected in 2 to 5 days. Results of postmortem toxicological testing are interpreted alongside the victim's history